Hi, I'm Arindam. I'm a doctoral researcher at the University of Eastern Finland. Today I'm going to discuss about my research on prediction of drug combinations based on efficacy and safety estimates. Drug combinations have emerged as a promising solution to treat complex diseases like cancer that usually involves multiple pathways for disease occurrence. Drug combinations in this case can be beneficial for targeting multiple pathways simultaneously, thus reversing the effect of the disease. Uh, Apart from this, they are also beneficial in reducing toxicity by lowering the dose required for each drug combinations. However, not all drug combinations are uh, effective and safe. Thus, there is a need for a robust method that can identify effective and safe drug combinations. Current computational methods for identification of drug combinations tend to focus um, on the safe on how good a drug combination will be rather than also focusing on if the drug combinations will have any associated side effects. Further, they also don't tend to be disease specific. Mm, Thus, there is a need for a computational method that can consider both efficacy and safety aspects for identification of drug combinations. Based on current research, we observe that network-based methods, a combination of network-based and machine learning-based methods has been uh, very promising for identification of drug combinations. So we thought of leveraging this and explore if we can use network-derived features for deriving efficacy and safety-related estimates for drug combinations to train machine learning classifiers for distinguishing effective and safe for distinguishing between effective and adverse drug combinations. We used a combination of random walk with restart and fast gene set enrichment analysis for the purpose. Starting from a protein-protein interaction network retrieved from StringDB, we try to identify the genes that are perturbed by each drug combinations. The top 500 genes that were identified to be perturbed by the drug combinations were then used for fast gene set enrichment analysis against three kinds of gene libraries. One based on efficacy, one based on safety, and the third one a combined library. For efficacy-related gene sets, we included all disease-related gene sets from different sources like open targets, PharmGKB, and um, theta, and these were based on different kinds of information such as literature based, differential gene expression based, or genetic based associations. For safety related gene sets, we included gene sets for various kinds of adverse drug reactions from ADREX. For the combined set, we included both disease-related gene sets and adverse effect-related gene sets. But we limited the adverse effects-related gene sets to only those adverse effects uh, that were known to be common for drug withdrawal. Once we have the normalized enrichment scores from the fast gene sets enrichment analysis, we use them to train the binary classifiers. We initially trained three kinds of classifiers: random walk with restart, elastic net, and support vector machines. Now, for training the classifiers, we did not have any gold standard datasets containing good and bad drug combinations for lung cancer and leukemia, that we used as a first case study. So we designed a dataset for training the classifiers based on information from drug combo DB and drug bank. The drug combinations that had at least one drug approved for lung cancer or leukemia predicted to be synergistic by drug combo DB and had no associated side effects in drug bank were considered as synergistic drug pairs. While drug combinations having at least one approved drug for lung cancer or leukemia and having reported adverse reactions in drug bank was considered as were considered as adverse drug combinations. Now we observed that for lung cancer, the dataset was highly imbalanced, consisting of only 31 effective against around 700 adverse reactions. In the other hand, for leukemia, we had kind of balanced dataset with 1,000 drug combinations in each category. So we used the SMOTE algorithm for balancing the datasets uh, prior to training the classifiers. The SMOTE algorithm uses a k-nearest neighboring based method to oversample the lesser class. We split the data set into two training and test set containing 80% of training and 20% of test set, and perform our 10-fold repeated cross validation for training the model. We observed that compared to the non-balanced model, imbalanced model, the balanced dataset performed much better. Further, we also observed that the random forest-based model had a higher area under the curve score compared to the other two, the GLMNet and the SVM-based models. Finally, in order to understand how well our method is placed against other tools for the similar other similar tools, we compared our tool to ProDeepSyn. ProDeepSyn is a deep learning based method for identification of synergistic drugs. We compared the synergistic scores from ProDeepSyn for lung cancer drug combination against our predictions, and we observed that the drug combinations that were predicted to be effective by our method had higher synergistic scores from ProDeepSyn's, but the drug combinations that were predicted to be adverse by our method had a distributed synergistic score. So though this is an initial comparison, we see that our tool may be better placed in identifying effective and safe drug combinations simultaneously, rather than the ProDeepSyn, which can predict synergistic drug combination but may fail at predicting drugs that might be associated with certain side effects. So with this, I end my presentation. Thank you.